Hi, good morning, everybody. Thanks to Oxford Nanopore for inviting me to give this talk. I'm really excited to talk to you about our recent developments in applying nanopore sequencing for cell-free DNA detection. Just as a brief outline of the talk, this is going to be a research study demonstrating the potential utility of nanopore sequencing for assessing cancer in cell-free DNA molecules. The first part of my talk is looking at something that's already been published as a proof of concept study. And second, the part of the talk is where we scale up our study for a larger cohort. Now, just as a brief introduction of why we want to do this. So breast cancer is typically detected using imaging-based techniques such as mammograms. And public health programs have actually been really good at improving survival outcomes in billions of women worldwide through early cancer detection. The earlier you can detect it, the better survival outcomes. However, there's still a couple of critical gaps that need to be met. For example, there's limited accuracy in early stage disease. There's a lot of false positives and false negatives with respect to having small lesions or lesions that have unclear interpretation. Also, there's limited accuracy in anatomic variation. So, for example, women with dense breast tissue often are diverted to things like MRIs, which are very costly and require lots of infrastructure. But lastly, there's a lack of equitable access to screening. So particularly in people in lower income countries or places that are more rural, don't have access to mammographic facilities. They have a lack of access to these public health initiatives that will really help them detect breast cancer earlier. So most people know about liquid biopsies, but for those of you in the audience who are, let's say, unfamiliar, lots of our group and companies and many academic labs are really interested in non-invasive measurements for detecting cancer itself. It is commonly understood as a platform where you draw blood from somebody and you look at something in the blood and measure it and see whether that is associated with cancer or not, and make a distinction of, yeah, you have something that needs to be followed up on. There are things like proteins that you can examine. Even in DNA, you can look at SNPs, their structural variations. Even circulating tumor cells, people have basically tried to extract circulating tumor cells and sequence them. But for the purpose of this talk, we'll be really looking at epigenetic modifications. What do I mean by epigenetic modifications? There are many of them across the human genome, but specifically, we'll be focusing on something called DNA methylation. What this is the addition of a methyl group, shown in there by the arrow there, on the fifth position of a cytosine through reaction of something called DNA methyltransferase. In humans, these are at CpG dinucleotide motifs. So basically, it's a CG, two bases. And they're scattered all across the genome, and they have some really important biological impacts. So for example, let's say we take a normal cell. You can imagine that DNA is splayed out as in the top row there. And you can imagine that a tumor suppressor gene is transcriptionally active. So tumor suppressors basically prevent a cell from going to malignancy. So as this tumor suppressor gene is actively transcribed, its promoter region has a pattern of methylation that is reflective of its underlying transcriptional program. As a cell progresses to malignancy, you can imagine a situation where the tumor suppressor gene is inactive, and this is reflected in an alternative methylation pattern in front of this promoter region itself. So basically, the question is, our main goal for the purpose of methylation-based cancer detection is to basically determine what are these changes in methylation patterns between people who are healthy and people who have cancer, and develop assays in order to study this. In general, there are about 30 million CpG sites across the human genome. So this is a lot of potential biomarkers that we can use for cancer detection. So traditionally, methylation is measured with short-read sequencing by something called bisulfite conversion, or you can use other enzymes to do this. What it does is you can imagine a situation where you have a series of a string of DNA. Some bases are methylated, some cytosines are not. The action of bisulfite converts canonical cytosines, which are unmodified, into uracils, which after PCR amplification are read as Ts. So this compresses 4 bases into 3 bases. So anything that you see that is remaining as a cytosine in your sequencing data is putatively a methylated base. Now, this has been generally successful in a variety of ways for analyzing methylations in human genomes, but there are a couple of problems with this approach. So But predominantly, one of the problems in applying this kind of analysis for cell-free DNA is that there is a wide extent of DNA damage that happens with bisulfite sequencing. There's also PCR amplification biases that may occur, so it's kind of a subtle point out in the previous slide, but when you apply bisulfite sequencing, you're changing the GC bias of the human genome, and this affects which fragments get amplified, which ones don't. So this needs to be corrected as well. Similarly, when you sequence CPG islands or when you apply bisulfite, you have sequence alignment issues, so they might not be placed in the correct portions of the genome, and this is a problem for analysis as well. And finally, when you do PCR, you always have to do complicated deduplication using molecular barcodes, and we prefer not to do that. So our group decided, well, why not use nanopore sequencing to look at cell-free DNA? So there's a bunch of advantages that we want to take advantage of in nanopore sequencing. First of all is that you get native methylation detection for free, essentially, in your sequencing data. You don't have to do PCR, so there's no deduplication. But really the most important part is that you can use machine learning models on your archive raw data, so your data sets become better and better over time with higher quality, and also you can start looking at alternative methylation marks that may not have been possible years ago, but are possible now. So with that in mind, let's look at one of the studies that we have published very recently, about 6 months ago, where we took nanopore sequencing and applied to cell-free DNA to see what we can ascertain from the single molecule profiles. So long story short, in this paper that was published earlier, we can sequence very tiny amounts of cell-free DNA and measure the methylation status of single cell-free DNA molecules. Here is a very simple plot of one of our first experiments. On the x-axis is the input amount in picograms, and the vertical axis is your sequencing yield. The sequencing analyte is basically leukocytes that we've digested with DNAs at open chromatin to mimic cell-free DNA's fragmentation profiles. The red line there is one of the official protocols from Oxford Nanopore, and our blue line there is basically optimizations that we've systematically improved in order to get over an order of magnitude improvement in the sequencing yields. This is with old R9 chemistry, so with Kit 14, this curve is way higher now, so we can get crazy amounts of sequencing data from very small blood draws and very small amounts of plasma. Moving on to actual patients, so these are a small cohort of about 20 patients with colon cancer. We can see that we get a very large range of sequencing yield. This is generally taken from about 1/2 ml of plasma, so it's about 1/10 of a tube of blood, so it's actually a very small amount of analyte that we need to use. And when we scale up, you can anticipate that this data will be at least 10 times higher. We can see that the sequencing reads go from, let's say, 1 million all the way up to hundreds of millions of reads. So the sequencing yields are pretty good for analysis. We can also look at the methylation profiles themselves. So this is a heat map showing the differential methylation between healthy donors and colon cancer patients. The top rows are 4 healthy donors, and the bottom is the 20 colon cancer patients we have shown. And what's shown on the x-axis is a series of differentially methylated genes that we discovered through this analysis. What's important to understand is that the healthy donors all have a generally pretty consistent methylation profile, and the colon cancer patients have a large range of different methylated genes, indicating either multiple types of colon cancers or there's a large transcriptional dysregulation, as reflected by these cell-free DNA methylation profiles. Lastly, in this part, what we can also see that's unique to nanopore sequencing is that you can get the native lengths of the actual cell-free DNA as it goes through the sequencer. In the top panel is a single healthy donor. What we can observe is mononucleosome peak and a dinucleosome peak, and also probably a trinucleosome peak. That's reflective of the actual fragment size of the DNA that's going into the sequencer. On the bottom panel is a single cancer patient. We can see that it is highly enriched in mononucleosomes. So this can also be a potential biomarker for detecting cancer as well. As we carry forward to the entire cohort, we can see this is highly statistically significant. So basically, the idea is that possibly when you have patients with cancer, you have more of an enrichment in smaller, shorter fragments of DNA. This could possibly be highly informative in terms of detecting cancers at early stage. So with this proof of concept, we felt pretty confident that we wanted to scale up to larger numbers of patients in order to build sort of a classification model for early breast cancer detection. And this is what we've done for the past probably like 2 years. I'm really excited to show you some results. This is basically all unpublished. So here we've enrolled 3 populations of case control. So case basically means patients with a confirmed clinical diagnosis of breast cancer. Controls is those who have a clinically confirmed, like know that they're just healthy. Here in total, we have about 835 participants in the study. And to my knowledge, this is one of the largest breast cancer cell-free DNA sequencing cohorts that's ever been undertaken, at least in the past, in the world genomics for methylation sequencing. In terms of the stage distribution, the vast majority of these participants are stage 1 and 2. So this is patients who do not have regional metastasis to distant organs. So this is really critical for detecting cancer, building models that would detect cancer at its earliest stages. We also have a small number of patients who have diagnosis of DCIS. This is a precancerous state, which is really interesting for detecting cancer even earlier than a confirmed stage 1. I'll also like to point out that population 1 and 50% of population 3 are drawn before treatment. So they have had no chemotherapy, no surgery, no radiation. This is really important for building accurate methylation models. So in terms of bioinformatic process, I also like to know we put all of these in the P24. So these are all Promethean. This is all Promethean data. We have over 50 terabytes of raw sequencing data. I think this is an underestimate. I think it's actually quite a bit higher than 50 terabytes now. This was processed with Stanford's high-performance computing cluster. We harmonize all the base calls using Dorado server 7.08. I think it's about a version behind now. This generates methylation calls, and we summarize everything with ModKit. The output of this that we'll put into our model itself is BAM files with modified base calls and bed methyl summaries, which is basically a tabulation of where every site in the human genome where we detected methylation or canonical bases. So this is one of the earlier plots that we've done in probably like last year in terms of doing QC of our samples. So we exclude samples based on sequencing yields, so samples that have very low sequencing amount of data, we just don't include analysis. This is a very normal part of dropout that occurs during the data generation. This represents almost less than 5% of our total cohort. We also do additional QC to make sure there are no sample mix-ups by taking leukocyte-derived exomes that we have from these same population, take their SNP profiles, and we match it to the cell-free DNA sequencing pileups, and do a maximum likelihood estimation of which cell-free DNA is likely to match with which exome, and we can confirm that we didn't have any large-scale sample mix-ups or any mix-ups at all, really. In terms of building a classifier itself, there's a couple of procedures that are involved in this. For those of you who are into like building machine learning stuff, this is really critical to ensuring good results. One is the training and testing scheme, basically which samples do you use to build the model and which samples do you use to test that it actually works. We use a method called leave one out validation, where we use one sample as testing and all the other samples for the training process, and we just iterate through. One of the reasons why we do this is that it enables us to generate a call for every single sample in our data set and to maximize the informativity by using more samples in our training process. For feature prioritization, what this means is which CpG sites do you actually use for the model? Because there's 30 million of them. That's a very, very large number of biomarkers. Not all of them will be informative, so we use statistical testing to narrow down this range. And the classification procedure itself, we use a random forest classifier in order to build a classification procedure that is not biased for different data sets. So basically, in terms of feature prioritization, we're asking which methylation features are different between case and control. So you can imagine a data matrix where each row is a particular CpG site in the human genome, and the columns are basically each sample, and the values in this matrix are the methylation value at that CpG site. You can simply just do a t-test between the case samples and the control samples to arrive at a p-value, and after multiple testing correction, you can then narrow this down to a smaller amount of CpG sites that are known to be informative and to carry forward for further analysis. So in total, there are about 30 million CpG sites in the human genome. We can narrow this down to about 500,000. So basically, it's we take 1% of these biomarkers and carry them forward for analysis. In terms of the classification procedure itself, we use a similarity score. So the way that this would work is you can imagine a test sample where for the CpG sites that we have prioritized is shown in that test sample in each row. The red is the hypermethylation, and let's say blue is hypomethylation. And we can directly compare to each sample in our case control data set and build similarity scores to say how similar is this one sample to the case samples and to the control samples. And these can become features for a random forest classifier. So I hope you can also see that the test samples, this test right there is matching the case. So this would be from, let's say, a patient that would likely to have cancer. Also, from a few slides ago, I've mentioned that fragmentomics is really important for classifying, as an informative feature for classifying whether a cell-free sample, DNA sample is likely to be from someone that has cancer. So we feed in features such as the length of the DNA, the amount of mononucleosomes, the amount of dinucleosomes into this model in order to improve the accuracy of the classification procedure itself. So before I go into some results, I'd like to basically remind everybody of the performance metrics that we typically use in classifiers. So the sensitivity is basically asking the question, how many positives do you get right in our sequencing, in the classification procedure? The specificity is how many negatives do you get correct? And the accuracy is essentially the combination of the two. What is the true positives and true negatives divided by all the samples in your data set? So okay, here are some results. So the output of a classifier is essentially a prediction probabilities, right? So given some certain cell-free DNA data, methylation data, and some fragmentomics, it outputs, let's say, it would give X probability that this is likely to be cancer. And basically, depending on where you choose your threshold is basically where you make this call. And this threshold is dependent on the actual needs and basically what your purposes are for designing this. So it could be for screening purposes or for diagnostic purposes, and those have different requirements of what you want for sensitivity and specificity. And obviously, when you change this threshold, there's a trade-off between having higher sensitivity or having higher specificity. So often there are different ways to report this, but one way to report the performance of this classifier is at this point called the Uden index, which is that the maximum combined sensitivity and specificity, this maximum combined sum of the sensitivity and specificity, and that's at that dotted line where that arrow is shown there. Obviously, you can move this curve around to have different reporting characteristics. But what's also important to note is that these samples are predominantly stage 0 or DCIS and stage 1 and 2. So this hasn't really been done before, and I would encourage you to look in the literature to see what the performance metrics of similar cell-free diagnostics would be in the context of breast cancer. I think we're doing really well in this area. These are some AUC curves. So on the left-hand side is the precision recall curve. Precision-based is essentially given a positive result, what's the likelihood that this is actually correct? And on the right-hand side is the classic ROC curve. What we can see is that the area under curve for both of these is roughly around 0.8, which is really good for a machine learning model. When you break this down by stage, you can see that it follows a classic curve of as you have more advanced cancers, the performance goes up. We see for stage 1, 2, the performance and sensitivity is about 70 to almost 80%. What's also really encouraging as well is that at DCIS or stage 0, this is a pre-cancer state, we have pretty decent sensitivity for detecting breast cancer. So that's it for showing some of the results for our classification model and our current efforts. There's obviously a lot of work to go. We're looking for further drill down for the selected biomarkers in the study in terms to see what kind of pathways are being enriched from this statistical feature prioritization. We also are actively sequencing new cohorts, so basically an independent validation cohort in order to validate that our model is correct and is working well. We also like to leverage other omic modalities such as structural variants or SNPs or other things to improve performance. Also building more machine learning models. So what I've shown you here is a random forest model. Obviously, a lot of people are asking, like, what about neural nets? This is something we're actively looking into. And also development of actually comprehensive methylation analysis for cell type identification to improve our model. So that's it. I'd like to acknowledge everybody in Hanli Ji's research group, in particular Shangxi Bai and Alison Almeida. They've been really critical for basically advancing this project and make sure everything is robust. Also, Lincoln and Brett at Intermountain Precision Genomics, who are really critical to this collaboration. Stanford Blood Center, Stanford Tissue Bank, and Stanford Cancer Institute as well, and all the patients who made this study possible. I'm funded by the NHGRI, NCI, Intermountain Precision Genomics, and also Clayville Foundation and Mark and Lynn Benioff. And if you'd like to get in contact with us, here's our contact information. Thanks.